And I can see that the numbers are going up very quickly. And we're going to wait one more minute and we'll get started. Okay, well, welcome. And Carlos, if you want to go ahead and start the recording.
It is going.
Okay, thank you. All right. Welcome to this afternoon's plenary on the last day of the conference. This is, unfortunately, the last time we're going to be able to meet as a group with all of us on the call. So I wanted to at least take one last opportunity to thank everyone, and truly for all their patience, their forbearance, the hard work that went into this. We pivoted within a matter of weeks to be able to go full virtual. And if it not for Sarah Miller and John and Shannon and Doreen Albertson from the ISIAC Secretariat, and John and Shannon from Syria, we absolutely couldn't have pulled this off. But we wanted to thank the ISIAC board and the president, Corinne Mandin, and all of our organizing committee and the chairs that have sat in as such, and the presenters, all of you that have presented, oral sessions, poster sessions. You know, you took time to take the training, to prep, to be ready, and we understand, you know, what a monumental effort it was on your behalf as well to make this a success. Also wanted to definitely thank Mr. Sean Hirono from Department of Health here in Honolulu, and for all of his time that he took to participate and try to make it work on site, which we did our best. And with that, Carrie Kenny, which, I mean, after this, I assume, Carrie, you're going to hibernate for maybe 8 months before you come back up. And
as long as it's warm, hibernation.
So, Carrie, I'm sure you have a few things, comments as well. Why don't you go ahead and
well, I just wanted to thank all of the people that you thanked as well. So I think you did an outstanding job on that. And also just for the whole community for sticking with us as we attempted multiple times to hold the conference. But I just want to comment on how amazing I think the content of the conference was, and also to remind you that we've been recording all of these sessions, so you can't be in 3 places at once. So you'll see that Tuesday's sessions have already been posted, and I even think yesterday's sessions have been posted. So as an attendee, you have access for that to those recordings and those other talks that you missed. So I invite you to go see those talks. Also, for both the authors and for the attendees, you can also leave questions if you didn't get your questions answered in a particular session. You know, we were in pretty We had pretty compact sessions. Feel free to leave a comment or a question for the author and authors and presenters. If you wouldn't mind going to check if you've got some questions there from some of the attendees, I think that would be great. So thanks everybody, and thanks to Richard, John, the entire team at Siri, and all the stage managers that have been helping us. They're the ones who are actually making this work. So thanks to everybody. And with that, I think we're ready to kick off. So back to you, Richard.
Okay, we're going to go ahead. A couple of slides I wanted to briefly go through, and if we could advance that, Carlos. We definitely want to promote and encourage you to be a part of the flagship conference, which ISIAC does every other year. This upcoming year, well, this year, Indoor Air 22, it's in Kuopio, Finland. And go ahead and advance it. It is June the 12th through the 16th. And the themes, you can see them here. I don't need to go through them, but they are all inclusive, and they certainly cover the entire cross section of topics that are current and what we're looking at in the future. So related to the indoor environment. Can we advance it again? Okay, so with that, unfortunately, Mervi or Pertti Pasanen, the president for Indoor Air 22, were not able to be on here, but they did send us the slides. This is Kuopio. It's kind of like an island in the middle of water throughout Finland. It's beautiful. Everything there is surrounded by lakes and waterways, and you got to remember you're upwards of 20 hours of daylight, and it's an unforgettable experience. And not only for the content do we want you to attend, but to again come and for so long, for the first time, be able to see each other person to person. So we welcome you to that on behalf of ISIAC. Go ahead and move to the next slide, and we're going to go ahead and start with our first plenary. And with that, Anita Van Breda. We worked with her for such a long time to be a part of a conference before that we did with ISCS, but she unfortunately had a conflict, but we're so fortunate to have her here. So a little bit about Anita. She's Senior Director of Environment and Disaster Management, World Wildlife Fund, Washington, D.C., and she has over 20 years experience in conservation and disaster management. In her current role, she leads WWF's work supporting environmentally responsible disaster recovery, reconstruction, and risk reduction, including international policy operations and training, and is a member of the WWF internal COVID working group. Anita has worked with a range of humanitarian, environmental, and development agencies on multiple disasters around the world, has a master's from the Yale School of Forestry and Environmental Studies, and an undergraduate degree in biology from George Mason. She is also co-lead of the United Nations Emergency Shelter Cluster Environment Community of Practice. So with that, Anita, so happy to have you here, and we'll turn it over to you.
Thank you so much, Richard. It is a pleasure to be here finally with you, even if it is virtually. I appreciate your patience in in getting me as part of this conference as well. And I have to stay. I hope I stay on your mailing list for future, because you certainly choose great locations for your conferences. So thank you also to the organizers and all the folks behind the scenes helping to make this successful. Really do appreciate it and look forward to chatting with you. Let me share my screen. Let's see, here we go. Okay, so again, it may be a little bit different for your attendees to be hearing from someone from World Wildlife Fund talking about the environment and disaster. So I hope that I meet your expectations and look forward to the conversation. But I want to start with talking about infrastructure and the amount and type of infrastructure that we expect to be built in the next few years. It's quite a significant amount. And if all this infrastructure is constructed over the next few years, it's estimated that will double the stock that we already have. So that is pretty sobering data to consider. And we in WWF, although we're known primarily for our species and wildlife work, we actually do a lot of different environmental type of work across different biomes and different issues. And we have looked at the issue of infrastructure and appreciate that it will have a bearing and an impact on all of our goals and all of the work that we do. It will impact our conservation as well as the sustainable development aspirations that we have working with our local partners and communities around the world. And when we talk about infrastructure within WWF, we have different types of infrastructure that we're looking at. So we look at human engineered infrastructure that is essentially meant to deliver environmental and social benefits. And these might be things like renewable energy, parks and protected areas. That might also cause some harm to the environment. Then we look at human engineered infrastructure that causes harm to the environment and the local communities, but also provides some benefits such as financial development, livelihoods. And these might be things like roads, dams, ports, and mines. And then we also look at natural infrastructure and how nature's engineering can provide infrastructure services to people, and how we can use nature, particularly in the context of a changing climate, for both mitigation and adaptation purposes. And that might be things like wetlands, forested slopes, sand dunes, etc. So the program that I lead, which sits in our climate goal, is focused specifically on disasters. And the goal of our program is pretty simple, and that is to use the environment and the services it provides to support disaster recovery, reconstruction, and risk reduction. And so we use the way that WWF does conservation and natural resource management and apply it to a disaster rebuilding context. And we're looking to do that for a number of different reasons. One, we want to minimize or eliminate the negative environmental impacts associated with that rebuilding, but we also want to improve the outcomes for the human communities that that disaster work is meant to support. So we're looking to support social and ecological resilience. And for us, that is the area where environmental management, development, and disaster management overlaps. But of course, we also have to recognize that all of that must be climate informed and speak to both climate mitigation and climate adaptation to the extent possible and appropriate. So we know that disasters are sadly increasing in scale and scope around the world. We know that climate change does fuel some of that risk and some of that impact. And the rebuilding that takes place post-disaster is often trying to rebuild in a number of months or years what may have taken generations to build in the first place. So that can represent a sudden and immediate and significant demand on building materials, their extraction, their creation, and their use. And so a lot of the work that we do and with my team is focused on infrastructure related issues. We also have a natural-based flood management program, and then we have a training program on how to rebuild and reconstruct using green practices. So these are just some of the events, the extreme events that we have been involved with over the years. It spans the globe and is growing again in scale and scope, including the last few years, of course, COVID-related issues as well as other hazards and disasters. And our experience includes issues such as emergency relief camps that are temporary, but where and how to place them so that they're safe, going all the way up to rebuilding community infrastructure, including homes, schools, hospitals, and those sorts of structures. Doing that with a range of different partners, including non-governmental organizations, as well as national governments, and then agencies like the United Nations. Now my screen is frozen. Sorry.
It
Okay.
advanced. Yeah, there you go.
Okay, great, thank you. So this is our green recovery training program. I've highlighted a couple of the modules in that program that I think are most applicable to the healthy building community. And we look at healthy building and particularly construction from the construction project cycle, but also the material life cycle. And we're working to support decision makers to make informed decisions about the trade-offs of these different issues in order to select and utilize appropriate building materials and construction materials. And so we've gathered some of our learning and our approach into our building material and environmental guide, or BMEG for short. This started after the 2015 earthquake in Nepal and was centered on that country, but we've expanded the scope of the guide to other countries, other partners, and most recently we partnered with Northwestern University with their engineering program, working with their graduate students and their professors to expand the number of materials that we address in the guide and expand the properties that are addressed. And Northwestern is working on an optimization tool to put this guide into practice. So in addition to trying to innovate and develop practices on the ground and in the field, if you will, that are practical and applied, we also tried to take that learning to a policy platform, either at the national, regional, or the global level, to mainstream these types of innovative practices, learn from others, but also make it more available. And so we're trying to expand the scope of and the type of partnerships that we're engaged with as this process unfolds. And so within WWF, as an environmental organization, we tend to look at the landscape and look upstream and downstream at what is happening in a particular place, what the environmental issues are and what the social issues are, but then funnel that down to a particular scale that is focused more at the household level or at the community level, but also going the other way. So we're trying to take a wide lens, but also be practical and applied for different application in different contexts. And in doing that, I think we've decided and aspire to use a healthy environment as part of a healthy building process. But particularly in the context of a changing climate, there's a lot to learn and there's room for a lot of improvement. So we've really been keen to focus on training the next generation of practitioners in the environmental field, in the disaster management field, in the climate space, in architecture and in design, and in the disciplines and sectors that are represented in this Healthy Building Conference. I think going through this, putting this together, thinking through your audience and the issues that you address, I'm going to take that back to my team to see if we can't expand our level of understanding and appreciation for the issues that you all address in healthy building. And I welcome the opportunity to speak with any of you about how we might collaborate as a sector and as a discipline so that we're more successful advancing these issues together than we might be individually. So with that, I want to thank you for your time. It's a very quick and short overview. But again, I think being invited to this conference, finally making it after a few years of missing out. Sorry to not meet all of you in Hawaii, but I appreciate the opportunity to look more closely at your sector and to understand the work that you do so that I can understand how I can work with my team on how we could benefit from your expertise and experience, but also would welcome the opportunity to share what we do and how we do it and see where there might be synergies for collaboration in the future. Thank you.
Thank you. I think we're Richard's coming back on. Oh, there you are, Richard. I was going to look for the questions.
I looked through them. I don't think we have any at this point.
Can I ask a question then? I work with a group that a humanitarian engineering group at UT Austin, and they actually have been working with the International Red Cross on products and developing new products and other things for refugee camps. And one thing I kept thinking about is that picture that you had that kind of struck me that, oops, there's something. It struck me that the tents that you showed, many times after a disaster do folks go to temporary lodging or refugee camps or something else, and it ends up being the semi-permanent housing? And is there a way to make that transition happen more quickly with local materials, or do you have any suggestions on that? Because it does seem after a disaster, you know, you might come in years later even, and you know, they might still be in what was considered temporary housing.
Yeah, thank you for that question. That is a critical issue, and it's a tough one to unravel, and it's a tough one to discuss for a number of reasons. It's all context specific. So
Yes.
it depends on the country. It depends on the approach of the national government in that country. And then it depends on the event itself and what makes most sense. I will say that over the years that I've been doing this, so since 2005, my understanding and engagement with the humanitarian and disaster management community has seen them change over the years. When I started in 2005, constructing at some level temporary housing was quite common in many instances. Less so now for a number of different reasons. Most people don't care for that kind of housing, and a lot of it does become permanent. So it depends if the context allows and people can be safe and secure while the more permanent rebuilding takes place. That's usually the more strategic and sensible approach to go. But then again, it just depends on the situation and the context. The question of building materials, and when I started, there was a notion that to be environmentally responsible, you should secure building materials as close as possible to cut down on transportation costs, which makes sense. But again, it depends. It depends on the impact of that local material, how it was extracted, how it was used. And that's a trade-off compared to maybe bringing in something from somewhere else that also has a bearing on local economies and livelihoods. So all of that needs to be factored into the decision-making and the trade-offs that are made. There's always a cost benefit, but that can be considered from different vantage points as well. And you raised the question about tents. there's work that groups I'm involved with now that are really trying to update guidance around tents and the disposal of tents and how to do that safe or to repurpose them. So looking at tents, the use thereof, how they're created, where they're stored, how they're transported, how long are they used for, are they healthy, are they the right material? All of that goes into the decision making. So I don't know how much your colleagues talk to disaster management groups and their procurement officers, but if not, I highly encourage you to do that, because again, I think disasters are increasing in scale and scope. There's more attention need to these issues, and I think your community has a lot to offer to that space.
Great, thank you. Just gonna look here. I have another question from the audience, or why don't you go ahead, Richard? Do you see the question?
Yeah. The question, are you involved in sustainable agriculture in developing countries, especially for small farmers?
Yeah, we have a whole team within WWF working on food production at different scales. That's not my team. That's not my area of expertise. That does come up in rebuilding livelihoods in a post-disaster context, which my team does work on. So yes, we have a number of different activities and engagement in that space. And the person who asked that question would like me to connect them with others in WWF, happy to do that. My email is available.
Right.
We have another question. Is there communication between different disaster management groups in different countries?
Yes, again, that also varies place to place and agency to agency. I put up the little logo of the United Nations Shelter Cluster. That is a group of like-minded, driven institutions, both government and non-government, academic individuals can be members of the shelter cluster. And that's really a coordination and a collaboration and communication body. I'm part of the environment community practice for that group. So yes, there are ways and opportunities for these different groups to connect with each other. Like any sector, that's hit or miss sometimes. And it's really difficult, obviously, to get to know colleagues in the middle of a disaster aftermath. So we also encourage people to connect in advance of extreme events. So if you do happen to meet each other working on a disaster response, you have some context and knowledge and trust to build upon.
I was going to say, are there any other questions? Oh, there's another one. This one's for you, Richard.
Okay. I don't see it, Carrie.
Then it's for me. Let's see. I just saw it. So you probably, oh, sorry, I'm seeing multiple questions here. So now they're flowing in too quickly to read. But is the question is the World Wildlife Federation already plugged into key organization considering world disaster response? Perhaps they could facilitate getting ISIAC reps involved, integrated into these discussions, and get more focus on healthy recovery.
Yeah, we're involved with a number of different groups, both formally and informally. I will, as my role in the shelter cluster, bring up this issue and happy to share. And yeah, happy to talk to anyone who feels like they'd like to participate or have more representation from your group in that space. I think that might be a valuable contribution. I'm not the manager of these groups. I'm a participant, but I can certainly help make those connections.
Okay, and I have one final question there, Richard. Other than the speed of construction, is there any reason your materials recommendations wouldn't apply to non-disaster response construction?
I don't think so. I think they do apply. But again, our key audience, our key partner, at least for my team, is that sector. So we try to take the materials and modify it and adapt it to the mindset, the funding, the milestones that group of individuals and agencies live with. Might be a slightly different context. For example, purchasing and the time frame for purchasing might be different in so-called normal development versus disaster response. But no, I think the principles and the approaches apply to both, and we try to learn from all the communities involved in this space.
Okay, well, thank you very much.
Carrie, why don't you, I know
Yes.
Greg Whiteley is there.
Yeah, so can you advance the slide, Carlos? Thank you. There we go. Hello. Yes, you match your picture, so we don't have any imposters here. So I'm pleased to introduce Greg Whiteley. Dr. Whiteley has a Bachelor of Applied Science from Hawkesbury Agricultural College, Masters of Safety Science from the University of New South Wales, diploma from the Australian Institute of Company Directors from the University of New England, and doctorate of philosophy from Western Sydney University. He's the chairman of Whiteley Corporation and an adjunct fellow in the School of Medicine at Western Sydney University. He's a collaborating partner study director for an IMCRC grant with the School of Medicine at Sydney University, which is investigating novel solutions for biofilm-mediated infections and other forms of biofilm contamination. And with that, let's hand the presenting baton over to Greg, and very interested to hear what you have to say.
Thanks very much,
Carrie.
Carrie, and thank you, everyone. I'll just
Greg,
pull up our slide deck.
for participants, if you could use the Q&A as opposed to back and forth between the chat, that will help Carrie and myself to be able to collectively look at the questions toward the end. And thank you, Greg.
Cool. And hopefully is that up and visible for everyone?
Yes, it is. It is full screen and everything.
Ah, wonderful. I don't know how I've done that, but that's a marvelous thing. All right. Well, thank you, everyone. And I'm going to change the pace a little bit and talk to you about cleaning and cleanliness. And I want to thank the organisers for giving me the opportunity to talk. All the references are obviously going to be included in the slide deck, and quite a few of the papers I'm going to reference are actually available as open gold access or gold open access. So let's see if we can get this to slide. Next slide. Why can't I get next slide? Slide page down. There we go. Well, first of all, let me talk to you quickly about cleaning. Cleaning is a process. Dr. Mike Berry, who's a former scientific director within the US EPA, wrote one of the seminal books in this field, Cleaning for Health. It's been out for obviously quite a few decades now. And it really has not been improved upon because the fundamental application of cleaning is that process of removing. But as an introduction, let me say that after all of the sophisticated science that we see, and I'm personally, I'm embedded a lot in healthcare and hygiene involving both surfaces and reusable medical devices. You would think that there'd be hugely complex ways of examining cleaning in healthcare in particular. You would be wrong. Unbelievably, for really the last 4 decades in particular, cleaning has been managed with visual inspection. So people go looking for microorganisms that they physically cannot see. It's quite ridiculous. And there are problems as to why we haven't progressed further, and I'm going to deal with some of those and some of the answers to that. Now, another thing about cleaning is you might find this hard to believe, there's actually no uniform definition of what constitutes clean. There are some specific examples, particularly in industry, there are examples. In parts of a hospital, there are specific standards, for example, in an operating theatre. But when you get out from that, the standards are actually almost non-existent. There are certain products obviously in therapeutic goods that have standards, but when it comes to foods and things like shampoos and different sprays, there are no microbiological standards that generally apply, and general cleanliness is not defined. Now, we've dealt a lot in the conference over the spread of pathogens. This little cartoon is from Dr. John Otter from a journal called the Journal of Hospital Infection. And of course, this is the standard way that bugs move. We've got an infected person. They're expressing all of these bugs in different ways. Some of them become airborne. Some of them are heavier droplets. Some of them are on their hands. We touch our face 20 to 40 times every hour, every one of us. And so our hands become mobile fomites and contacts for disease. We all shed 40 million skin cells a day, and the flakes are just falling off us. We're all like little pig pens roaming around the place, dirtying everything up. And as a consequence of that, the bacteria that live on us and in us that we actually need to survive, they fall off as well. But in particular contexts, that can become a problem. And in my area, which is particularly in healthcare, that's a really significant problem, because we then match that with microorganisms that do not respond to antibiotics. They resist antibiotics. Now, the two primary means of spread, obviously you've been dealing a lot with droplets. I mean, that's obviously a core part of the process with COVID-19, was working through from the emotion about it's on every surface to realizing it's an airborne spread. It's a cold virus. And as the pandemic rolls into its third year now, and I know you've had a lot of talk about COVID, we've really got to understand and had to come to terms with it's a virus that's spread primarily by aerosols. One of the funny things about this virus, of course, is it actually does survive pretty well on surfaces, and many viruses do. But we're worried in cleaning about more than viruses. One of the quite extraordinary things is that whilst there's been a huge number of people who have died globally, it's been completely tragic. The superbugs that are inhabiting hospitals have not been on holidays, and we've still had vast numbers of people die from multi-drug resistant organisms. And the work that we've done, particularly around biofilms in medical spaces, has somewhat been seminal. So one of the seminal pieces of work that we've done is to show that whereas the old way of thinking was that many of these bugs will actually die quite quickly on surfaces, in fact, particularly bacteria, when you put them into a biofilm that's been allowed to develop naturally, a lot of these microbes, particularly the superbugs, will drop back into what's known as a persister cell state. And we've shown and published that they can actually live for years. So surfaces that have been untouched for long periods of time, you might think, oh, all the bugs are gone. Whereas in fact, if they're in a biofilm, they are still there and they are still potentially viable and culturable. What's more, if you touch an active biofilm, and the little red agar plates on the top are from a paper by one of our Ph.D. students, we showed that this was using Staphylococcus aureus, that the bugs will continue to stay on your fingers for up to 19 subsequent touches. So if you touch a contaminated surface, those bugs are on your fingers for 19 subsequent occasions. Now, if during that 19 touches, you touch your nose or your ears, the bugs have moved from your fingers elsewhere onto your body. Likewise, another student of ours, Dr. Tahir, did work using gloves, and we looked at 3 different sorts of gloves. Do the gloves make a difference? Well, no. And in fact, what we found was that the gloves almost mirrored the handwork. That is to say, on the right sort of gloves, the bugs will stay on the gloves for up to 19 subsequent touches. Now that's very significant when you think about how bugs move around. And of course, one of the problems we have is that the spread of microbes is not necessarily uniform. Obviously, there are droplet patterns, there are sprays, there are particular material spreads. And I've got this slide up particularly to illustrate one critical thing. Glass is great for showing this. Obviously, you've got a little wash me sign on someone's window. You've got someone using a microfiber cloth to clean off the window, the soapy window that's going to be cleaned because they're doing it completely wrong, but we won't get into the technical aspects of that. But on the windscreen on the right, what you see is smearing. That is to say, singular clumps of dirt have been wiped across, and rather than removing the soil, it's been uniformly spread, or not uniformly spread, but certainly spread across a surface. Now, when it comes to cleaning, that is hugely significant, because if you use the wrong form of cleaning and it's done badly, and you've got clumps of soil, whether it's with biofilms or other materials, in hospitals we worry about things like blood, vomitus, fecal material. But of course, you get outside of those contexts and you move into a commercial or domestic building, and even in bathrooms with things like molds, you really want them removed. You don't want to spread them across a surface. Now, you would think that wiping a surface would be something quite simple. I had a run-in with a particular editor of a journal at one stage who was a nurse who said, all you need to do is wipe the surface. There are actually 9 separate variables in the simple act of wiping. So if you've got to control a variable in wiping, which one do you control? Now, what we've found is we've had to step back out of doing things in the field to go back into the laboratory. And we're certainly developing methods, and I'll show you one piece of data in a moment, on how we go back and re-understand how we're doing cleaning so that the cleaning doesn't act as the means of spread from these bacteria or viruses from one surface to the other. Other work we don't have time to deal with today has shown that, for example, with standard microfiber cloths, if you pick up material on the first surface you wipe that's got any one of a half a dozen viruses that will survive on surfaces, that those viruses will continue to fall out on every subsequent surface on which that microfiber cloth is used. Now, when it comes to biofilms, and let me stop for a moment to talk about a little bit of history, personal history. When I did microbiology as an undergraduate, which was a little while ago, we used to use test tubes, and it was a bit like sending the microbes on a holiday into the South Pacific. Everything's warm, there's lots of feeding and breeding going on, and they're all having a happy time. What we found is that when you move into the real world, in fact, the microorganisms can do an array of different things, but they love creating biofilms, that is, little places for themselves to live in. And when they do move from what we know as now as a quite happy state in a test tube into the sessile state of a biofilm, there's a whole pile of genetics that take place as the transfer from one state to another goes on, and the expression of genes lifts by orders of magnitude. So these biofilms end up being complex communities with a whole array of different things happening. Typically, a bug in a biofilm, Staph aureus, vancomycin-resistant enterococci, pick any one you'd like. Another great one that's a nosocomial infection is Acinetobacter baumannii. These bugs, once they're in the biofilm, are 200 to 1,000 times more resistant to cleaning and disinfecting. And the same for antibiotics. If they're a biofilm in a wound, they are 200 to 1,000 times more resistant to antibiotics. So in this little experiment here, which is published in 2019, we took a standard Staph aureus culture and dried it onto a surface and saw how quickly it was removed. And we managed to show that after just one wipe with a microfiber cloth, you would remove 3 logs, that is 99.9% of the bugs. But if you take that same Staph aureus and move it out of its standard growth state and put it into a dry surface biofilm growth state, There's a little bit of microbiology to do that. What we found was even after 50 wipes, you were only removing just over 1 log of the culture. So they are a lot more resistant, and that has massive implications for cleaning. And here's an illustration of what goes wrong when you don't clean. This is from a paper, Andy Lee et al. From Infection Control Hospital Epidemiology. In this study, which was in an intensive care unit in a public hospital in Australia, they were tracking a multi-drug resistant organism called vancomycin-resistant enterococci. And the patients were all coming into the ICU and were being monitored as they came in. The cases on the left are the cases where they came into the ICU and they found the bug. Now we're going to start with number 5. And as you can see, case number 5 quickly gave the bug to another couple of people in the ICU. But as things move around, and all the green dots in the middle of this slide are actually environmental objects. They're things like infusion pumps, the hover mats, the IV poles, point of care instruments, and a range of different things. And using modern genetics, we're able to show, or the authors were able to show, that the bugs that were on each of these items were specifically linked to the bugs recovered from the patients. But where it got really terrifying, because this was done over a 20-week period, over 108 days, was that we could then show that the bugs moved directly from these inanimate objects into subsequent patients. And the yellow patients on the right-hand side are those that were colonized, so they don't have an infection, but they have been colonized. The patients in red are those who have an infection, and the patients in the red squares are patients who are deceased, not necessarily due to the infection, but certainly died with the infection. That's what happens when cleaning fails. In hospitals, we do the best thing we can possibly do for the bugs. We gather our sickest people together. We put them in a close location with people who don't wash their hands and in places where surfaces are not properly washed and cleaned regularly. And patients die. Now, once you get one of these MROs, depending on the bug, will depend on the mortality rate. Things like VRE have a typical mortality rate following bacteremias between 25 and 33%. So how do you monitor the cleaning? And it becomes pretty important, not just in hospitals, but in around the place. Obviously, we've seen a huge array of ridiculous hygiene theatre with the COVID-19 pandemic. But when that's all subsided and all the emotions gone away, we still live with these microbes. And most microbes are of no problem to us. In fact, we live with microbes. Most of them live on us and in us, and it's all fine. But how do you monitor the cleaning? Now, in fact, there are some scientific methods that are out there. We've got 8 listed here. The common ones are the first 4. Visual inspection, microbial recovery, which is the problem with microbiology is it takes too long. You take 48 to 72 hours to get your answers. And then you've got things like fluorescent markers, which we don't have a lot of time to deal with. The real technology there was invented by a colleague, Professor Philip Carling, who's in Boston, and developed a product called DASO, which is a little marker spot. But then there's also this thing called ATP testing, which many of you will have heard of. ATP is an enzyme that is present in all cellular life forms that respire. So everything that breathes uses ATP as part of the Krebs cycle. And it's a marker, a surrogate marker for cellular presence or contamination. Now, there are some issues, and I don't propose to go through these slowly, rather just skim over this. The great advantage, which is at the top, is that it's a simple real-time test with digital results, and it gives you a quantitative answer. But the problems are sampling, inherent variability, and then how do you collate the data meaningfully? So here's a summary of the different benefits and difficulties. On the left, you can see the benefits. On the right, you can see the difficulties. And we'll start with relative scale, move to imprecision, look at variability, and then look at sampling methods. Of course, there are shortfalls there's been people out there in various parts of the world saying, well, you can use ATP to indicate for COVID. Well, you can use ATP testing to indicate for cellular presence. Viruses aren't cells. They don't have any ATP. They don't respire. If they're in a cell and there's cellular contamination, you'll pick that up, but you won't pick up COVID. Now, in experience that we published some time ago, we took a bunch of commonly used, the three most commonly used ATP devices that we could find. And initially we had 3, we've expanded it to 4 now. But what the first thing we found was that the scale is called relative light units. And unfortunately, it's relative in every sense. And there is no common understanding of what that actually means. It doesn't quantitate back to a real measurement of ATP. So every device brand has its own interpretation of the scale. And as you'll see, this is a log linear scale, and you can see on the y-axis scaling, some of these devices will read to more than 1 million RLUs, whereas something like the Hygiena unit will top out at just 10,000. And as you'll also see from this little graph, there are significant problems at the lower end of the scale, which is exactly where we want to measure cleanliness, because the lower limit of detection and the lower limit of quantitation vary dramatically between devices. And if there is any variance, you can't detect it. Likewise, at the top end of the scale, the upper limit of detection and the upper limit of quantitation, when you get close to the top end, these devices flatten off their readings and you can't tell. Now, that's probably not relevant because we really don't care what happens at the top end. It's clearly dirty and it's got to be cleaned. Now, when we looked at variance, and we used the coefficient of variance as a measure, for those that will remember their engineering science, it's the opposite of the signal to noise ratio, where we put in this case, coefficient of variance is the standard deviation over the mean. What's really terrifying with this is how much variance the devices showed. The black dots at the bottom are a calibrated liquid chromatography device, HPLC, or an LCMS HPLC. And as you can see, very little variance on the HPLC, but with these handheld devices, huge amounts of variance. In fact, there's so much variance that we showed with the paper I'm about to put up next, that any single ATP reading has a 20% chance of being wrong by a factor of 2. So there's a 1 in 5 risk that your reading, if it says 100, could be 50 or 200. If it says 1,000, it could be 500 or 2,000. Now and there's no way for a user to detect that in their use. We used a range of different microbes along with quantitated ATP to check out the variance. We use Staphylococcus epidermidis, we use Pseudomonas aeruginosa. Again, there's 4 brands there. And again, the variance is huge. What's really terrifying is where we get a variance where the standard deviation is higher than the actual mean. Now, when it comes to using these devices, and they're particularly widely used in healthcare, and they're being widely used in things like disaster restoration and other specialty cleaning areas, unfortunately, you are then giving a quite complex, inaccurate device to people who really don't understand data science very well and are not trained in classic instrument science and validation. And they go and start measuring things. Here's a range of different things that you might measure in a hospital: taps, handrails, door handles, wheelchairs, stethoscopes, mice. And of course, when you go back to the literature and go back to the device manufacturers, they're recommending swabbing areas of anywhere between 10 by 10 down to 2 by 5. But what happens with those irregular shapes? How do you do a 10 by 10 on a tap? I mean, it's quite ridiculous. And if you've got a 1 in 5 risk that the reading is going to be rubbish, that makes things even more complicated. So work we've done and published, this is up on the web on open access. We produced an algorithm where we did a number of things to transform the way we collected the data. This has been used widely. Here's one of the papers that was published just recently, using it in a very broad cleaning study. This is up on the web. The method that we originally published is on the left. It uses duplicate samples. That was basically adapted to the REACH paper. But here are the key features. Instead of just taking one sample, we've realized, and there's pretty good statistics on this, that if you take paired duplicates, you solve some of the inherent variability issues, and I'll go on to show you how. We also realized that you can then test out the cleanliness level and nominal cleanliness level in a different way. You can validate the data with a cleaning step, and then you get validated data as an output. Now, the cleaning step's really important. We'll come back to that. But here's some more data that we've published. Using a cleanliness threshold, this is using a hygiena device in a published paper from the American Journal of Infection Control. You get really nice separation on a risk framework where you separate out the dirty from the clean. The two equivocal groups of the quadrants here are very interesting, because what happens when you're taking duplicate samples, matched pairs, is that sometimes you'll get one sample of the pair will be below the threshold and one sample will be above the threshold. So we revised the algorithm last year after a couple of studies. We've published about 7 or 8 studies now. We realized we can simplify it, and I'm just going to skip past this to show you it's now a very simple process. You take your samples, you do your intervention step, and then you do your classification. It's quick, it uses more swabs, and we're using 3 swabs in place of 1, but the data benefits, as you'll see, are enormous. Here's some tables taken straight out of the recent publication. It's literally, I've got it there as 2021, but it literally is in this quarter's issue of Infectious Disease and Health. And there's some examples of the tables. You end up with really nice data. You've got good separation. You can count in normal numbers, but then the statistics are also measurable. But you end up with an array of different things that you can see. Again, you classify your dirty, you classify your clean, and you get this group where you're not quite sure if they're dirty or clean, because you've got one reading that says it's clean and one reading that says it's not. And from a risk view, that's really a great place to be, because it allows us a nuanced series of approaches. And in fact, getting matched pairs in what we've experienced is more important than the actual area that you use. As long as you're matching your pairs accurately, it's a far more reasonable statistical way to do it, and allows for much better pair-to-pair comparison. The other thing the cleaning intervention does is it allows you to test whether the surface actually can be cleaned, or what the level of expected cleanliness should be, because some surfaces are really hard to clean and you're not going to get to 0. So what does a very clean surface look like in a quantitative manner? And then this also allows you to do longitudinal studies. So let me give you an example. Here's a cold water tap. If you're going to take matched pairs, you use your first swab on one side, on 2 of the handle edges, and then you use your second swab on the second side. Now, this is data that I've partially taken out of a study we're working on and partially added some more data into. But you can see you end up with duplicate samples initially. You can then average those to give you a number. You then get to classify them, and I'm using a pretty standard color-coded approach of green is good, yellow is danger, and red is bad. But then you record your cleaning intervention numbers, and you get a very different series of numbers straight away. And what that allows you to do is move into really quite nicely separated tabulated data. Before and after data is good. I've got the initial reading for the clean versus the initial reading for after. You can do statistics, so you end up with standard statistics. I've used both Wilcoxon and Mann-Whitney to do the analysis. We are working on an app that we recommend, we'll be recommending. It'll be out in February, possibly March. Future work, well, we're working with Dr. Shaughnessy and Professor Havaninen and a bunch of other people doing a whole pile of work in this area. to improve our understanding of what cleanliness really looks like, and acknowledgements, and I hand back to coordinators.
Thank you, Greg. Carrie, you there?
Yes, I'm here. I was just seeing if we had any questions. We have a few minutes for questions before our next session. If anybody
I've got
has
a-
questions, if not, go ahead.
I've got a brief one, Carrie,
Go for it.
while we wait on if others are out there. Greg, you may not have been able to tune into the entire conference, but there have been several remarks of over cleaning, stop cleaning. Um, you know, is there a need to clean? Concerns about over cleaning, which are right. You know, certainly you've got to respect, but you know, um, I sometimes wonder if some people talking about cleaning have ever been in a U.S. school. I know, Carrie, you have, and seen the typical level of hygiene in there, which is in such need. And certainly there needs to be attention to the type of cleaner and frequency, but even the CDC goes on to say that in areas, high traffic areas with a large number of people that may be poorly ventilated, more than cleaning upwards of once a day or more may be necessary. How do you feel about that?
I think you're muted, maybe? Greg?
There we go.
There, go. Start again. We didn't hear that.
No, that's all right. Look, there are a bunch of questions in there. The first thing is it's over cleaning if you're doing it unnecessarily, and you've already done the job and you're just going over the same, and that's just wasting time and money. Hygiene theatre is where you've got people doing stuff that looks spectacular, but they're actually not doing anything. So people wandering around in spacesuits with respiratory masks on or full self-contained breathing and then spraying rubbish everywhere, that's not cleaning. So I think we need to define what cleaning is, and that's part of the problem. One of the issues here is this is a huge sector. The head of ISSA globally estimates that the amount of money spent on the cleaning sector in a global sense is somewhere above $2 trillion US. It's an enormous sector. And of course, there's also some evolutionary stuff about cleaning. Believe it or not, we actually are primed to get things clean because we know that if you don't have cleanliness, you can have a spread of disease. And in schools, certain areas are more particularly at risk than others. Obviously, bathrooms, if the kids don't wash their hands and you get someone who comes to school with a norovirus, you'll end up with 35 kids with a norovirus within 48 hours. Likewise, when they come in with influenza, well, the current thing, of course, is COVID. Kid comes in with COVID, you get a cough and splutter everywhere. It's only going to last a few days for most kids, but the virus is going to move around. Likewise, if you've got a kid who's going to a cafeteria and they've got some sort of a food poisoning bug or a gut bug, again, noro's the classic, then you really want to focus your cleaning. When we get into the classroom, of course, cleaning also includes things like not just the microbes, but dust, and there's an array of different things that you need to clean for. Mold will grow on dust. Bacteria will grow with mold. Cleaning is the process of removal. I think the question therefore is how do you do the monitoring? And that's what we're really focused out on. How do you do the monitoring? What represents effective cleaning without wasting money? Because the biggest waste of money is you pay for cleaning that then is done badly or not done at all. I mean, that's the most stupid use of cleaning.
I see
Thank you,
John
Greg.
is, John, do you have a question representing Siri in the cleaning industry? Do you have some thoughts here?
I don't really have any questions. I certainly, as Dr. Whiteley knows, I certainly concur with what he's saying. And really the key is, for me, is the last comment. The problem with cleaning is cleaning that doesn't occur, but is being paid for, or ineffective cleaning. One of the things that Mike Berry focused on a lot, and I know that Greg knows this as well, is high performance cleaning. If cleaning is done well, then you actually don't spend more money on cleaning, you spend less money on cleaning because you're actually removing the contaminants. But great presentation, Greg. I thought.
Yeah. Well, one question that's turned up is about COVID disinfectants and the EPA list N, which is a great question.
Yes.
Look, let me make a comment in two parts. Firstly, about the virus, COVID. It's actually quite an easy virus to kill on the surface. We're about to publish some data on an array of different things that work and don't work. There are some chemistry reasons for that. The COVID disinfectants list have been not always tested. It's a slightly different way in the US, but look, that virus is not really a big issue. And if you're cleaning it away, that's even better still. Some of the other bugs that we're worried about, particularly in infection prevention, are much harder to kill than COVID. Noro is a hard virus to kill. You know, we're all worried about some of the other multi-drug resistant organisms, some of which are very hard to kill. Things like Candida auris, which is a yeast, very hard to kill. And so list N is important, but it's more than about COVID. So.
I have to say it feels like COVID all the time now, but I do like your, what did you call it? Hygiene theater.
Yep.
Hygiene theater.
Yeah. But I think to John's point, you know, effective cleaning is really what we want everybody to be able to do. So I see that there are, with chemistry, there's another question here, and then we'll probably need to wrap up. We have to remember the quality of the wipes as well. I noticed you mentioned about the wipes. Is there, beyond, you know, what chemicals you're using, is there something about one wipe being better than another? You made this comment that you can basically use wipes to spread it around. So now I'm wondering, what are we supposed to do? What are the cleaners supposed to do? Do you have an answer to that?
Well, here's one of the ironies. If you talk to the companies who are selling all these products, their products are fantastic. But there are no standard methods to prove they work. So where you've got no standard methods and you don't have a defined level of cleanliness, it's really difficult. One thing we tend to promote with wipes is they should be used on one surface only. You should use only that one wipe on that surface, or let me rephrase that. You use only one wipe on one surface and you use it in one direction. If you're using a wipe and going round and round and round, you're spreading the germs around. And, you know, if you look how people clean a window with a squeegee, window cleaning is a great example of how to clean. You start at the top and you move in a very logical manner and you end up with a clean window. And the same applies on surfaces. The difficulty we've got is not everything's glass and the surfaces are not regularly shaped. So you can clean properly. As a microbiologist, you know, we do a thing called aseptic cleaning. It's a very specific type of cleaning, and we measure that we get the right outcome. But if you tried to do that in general community sort of ways, the cost would just be out of proportion. In terms of the wipes, look, we don't recommend any single wipe. Some microfiber is better than others. Some cloth wipes are better than others. Some paper toweling is better than others. Ultimately, what you need to do is clean and make sure that you've measured that you've got cleanliness.
Got it. And so that's the ATP and other readily available
We're
techniques.
actually carry a session coming up on schools where there actually is some insight into health outcomes related to roles of contamination. So there's a lot of work in that area.
Yep.
Yeah,
Thank
I
you,
think
Greg.
other than you've convinced me again not to go to the hospital, but I absolutely, you know, you could say don't go to the hospital, don't go to the university, don't, you know, we all have done the quarantine, essentially isolated home thing. So we're all over that component. So I think we're willing to go out again as soon as Omicron passes, and hopefully COVID-19 as well. But I just want to thank both of our speakers. We're about out of time now. This is going to be the speaker ready room for the next sessions. We have a couple sessions, one on schools that Richard mentioned, and then also one on thermal comfort and perception. And those are both sets of oral sessions. And then we have one more poster session. But I want to thank both of our speakers, Greg and Anita, for their excellent plenaries today, and also all the speakers and all the sessions, and all the Siri team and all the stage managers who actually made this happen. We're able to pull this off. So thanks to everybody. And this is the end of this session. So take care. Thank you. And stay out of
Thank
the
you.
hospital,
Thank
and
you.
no more disasters.
Thank you.